Good afternoon. I'm Dr. Brian Shapiro, a technical writer at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Angio-Ready, an improved in vitro model of angiogenesis, presented by Mr. Kevin Grady and Dr. Chaozong Zhou. Mr. Grady is the Product Line Business Manager, while Dr. Zhou is the Senior Scientist at the ATCC Cell Systems. In this presentation, Dr. Grady will introduce the Angio-Ready Angiogenesis Assay System, an in vitro co-culture system for measuring angiogenesis. Dr. Zhou will then present data showing that this GFP-expressing co-culture system forms functional tubular vascular structures and responds appropriately in a dose-dependent manner to known agonists and inhibitors of angiogenesis. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. Any remaining questions, as well as the recorded webinar presentation, will be archived on the ATCC website, www.atcc.org. Before I turn the presentation over to Mr. Grady, I would like to provide a bit of background information about ATCC. Founded in 1925, ATCC is a private, independent, nonprofit organization with headquarters in Manassas, Virginia. Our core mission is to advance and apply scientific knowledge. This is done through a multifaceted approach that includes the acquisition of materials which are either deposited or identified as a unique means of supporting scientific needs, the authentication of materials through a polyphasic approach that employs genotypic, phenotypic, and functional analyses, the preservation of materials as low-passage stocks, and the development of both acquired materials and novel scientific tools that have been generated at ATCC. The products at ATCC are also standardized so that we can provide resources for generating reproducible research data. Additionally, these materials are stably distributed to both domestic and international scientific establishments. Through these efforts, ATCC has been a source for the standard use of materials in vitro biological research for 90 years, adding further credibility to scientific discoveries. So with that, I would like to welcome Mr. Grady.
Thank you, Brian. So in today's presentation, I'm going to discuss the various methods currently used for angiogenesis assays, and then introduce ATCC's new assay system, Angio-Ready. I'll then turn the presentation over to Charles, who will discuss the features and benefits of the new system, and then discuss some application data and how you can use Angio-Ready in your current workflow. But first, let's start with some background on the angiogenesis process. First, angiogenesis is defined as the formation of new vessels from pre-existing vessels. This happens by the activation of endothelial cells by growth factors such as VEGF and basic fibroblast growth factor. Next, the capillary wall is degraded by extracellular proteinases, which then create a branch point in the vessel wall. This is followed by migration of endothelial cells into the extracellular matrix towards the angiogenic stimulus. The endothelial cells then reorganize to form tubules with a central lumen, followed by interconnection of the new tubules to form a branch network. So you can see easily how angiogenesis is a critical process for tissue development, wound repair, reproduction, and tumor development. One of our main goals in developing our new assay system was to provide an assay for the easy screening of new cancer therapies, which we will discuss in greater detail later in the presentation. Angiogenesis models have been around for many years. The first models, which are still in use today, make use of endothelial cells, most often HUVECs, seeded onto a matrix of some sort. Collagen and fibrin matrices have been used in the past, but most researchers currently use a gel matrix of something like cell matrix from ATCC or Matrigel. The tubes formed in this method are generally short and homogeneous in appearance, and are likely not representative of the true in vivo formation. This is a time-lapse movie of angiogenesis occurring after aortic endothelial cells that stably express GFP are seeded onto cell matrix. Notice the spiderweb appearance of the tubules with little branching and interconnecting. Please keep this image in mind to compare with our new assay that will be shown in a few minutes. So as technology advanced, researchers improved upon the original assay setup. In vivo, fibroblasts are responsible for laying down the cell matrix structures. So why not co-culture endothelial cells with fibroblasts to get a more in vivo-like assay? They did this, and they observed more heterogeneity of the tubules with more interconnections between the short and long tubules. This allowed for a more in vivo-like assay. But ATCC wanted to do better. The question was, how can we develop an assay with better reproducibility, ease of assay readout, and make it more biologically relevant? First, we started with our portfolio of H-chart immortalized cell lines. These are primary cells in which we have modified the telomerase gene such that the cell is no longer senesce, thereby having the growth characteristics of a continuous cell line. The modification is subtle and direct, so primary functioning is not affected. The cells are stably selected from a single clone, so there are no issues of donor variability like you find with using primary cells. You can only get a finite number of primary cells from primary tissue, so if you're running a large screen, you need to make use of multiple lots of cells. You're going to be introducing variability into your screen. But H-chart cells are derived from a single isolated cell, so every lot of an H-chart cell line will be identical and confer identical performance. So using H-chart immortalized cells conveys the assay with a high level of reproducibility. To make the assay have an easy readout, we took an H-chart immortalized endothelial cell called TELAHEC, which is an immortalized aortic endothelial cell that stably expresses GFP. So using this endothelial cell in angiogenesis assays makes it very easy to track tubular development with live imaging, requiring no need for fixing or staining. To make it more biologically relevant, we replaced fibroblasts with mesenchymal stem cells. Remember, mesenchymal stem cells are multipotent, so they can form various cell types related to angiogenesis and endothelial function, as well as vessel structure. Down at the top of the slide, the phase contrast slide shows the tubules growing on and among the MSCs. The GFP allows for easy visualization of the forming tubes. Alpha-smooth actin is a marker for smooth muscle cells. The blood vessels have a layer of endothelial cells lining the vessel lumen. The endothelial layer is surrounded by a layer of smooth muscle cell followed by fibroblasts. In the second slide, we stained for smooth muscle actin with anti-alpha smooth muscle actin, and you can see a similar pattern of fluorescence as seen for the GFP. In the last slide, we merged slides 1 and 2, and you can see that the smooth muscle actin appears to be expressed by cells surrounding the endothelial cells. The smooth muscle actin cells are likely smooth muscle cells arising from the MSCs in co-culture with the endothelial cells. Thus, we have created a more in vivo-like model for studying angiogenesis. This is a movie of our Angio-Ready Assay System, which is a co-culture of H-chart immortalized endothelial cells and H-chart immortalized mesenchymal stem cells. Notice the extensive branching between long and short tubules compared to the gel matrix model seen earlier. Hopefully from this movie, you get an appreciation of our co-culture method of H-TERT immortalized cells grown with H-TERT MSCs. We believe that this model offers the best representative of a true in vivo setting. So please remember this system, remember the image as you go forward through the presentation to compare the long branching interconnecting tubules to the short spider web appearance of the earlier models. The Angio-Ready system consists of a cryopreserved cell mix of H-TERT immortalized telehec GFP cells and mesenchymal stem cells. The ratio of the two cells is proprietary. And also in the kit, in the system, you also get Angio-Ready Angiogenesis medium. We provide the material in 2 packs, which is enough for 2 96-well plates, or a 10-pack, which is enough for 10 96-well plates. The 2-pack contains 2 vials of cells and 1 bottle of media, while the 10-pack consists of 10 vials of cells and 5 bottles of media. The media can be purchased separately in case something goes wrong during the assay, like a spill or contamination. However, the cells cannot be purchased separately. Now I'll turn the presentation over to Charles, who will provide an overview of the assay protocol and then discuss various research applications for which the Angio-Ready system can be a beneficial tool.
Thanks, Kevin and Brian. Good afternoon, everybody. I'm Charles Zhou, a senior scientist at ATCC. I am the developer of this product. Kevin has told you the benefits and the features of this product. So here I'm going to tell you how we are going to use the product and under which circumstances this product can be used. This slide shows how easy these assays would be. So it's just like you saw the cells, resuspend the cells in the media, and then feed it into the assay plate, such as a 96-well plate. Then after 18 hours, you can begin your compound screening. So this product is designed for a 96-well format, but we can also do in other surface plate if you, according to your own purpose. Such as here is some the listing of the plate we can use, such as a 96-well plate, 24-well plate, 12-well plate, a 6-well plate. Just feed accordingly to the protocol recommended. So next several slides, we will tell you about under which circumstances the product can be used, because we have several feedback from the customers how we will use this product for. So angiogenesis process in the first one is the angiogenesis process in the wound healing and regenerative medicine screening. So this slide shows a typical wound healing process, such that the wound will be filled with blood clot and the neutrophils invaded in the clot. At the second stage, endothelial cells will enter the clot and begin forming new blood vessels. The third and finally, the fibroblasts move into the wound healed, and the wound is healed. So here you might want to ask, what are the promoting factors for the new vessel formation? So if you have a compound candidate, is there any easy but reliable screening method available? The answer is yes. This slide, we all knew that VEGF is promoting angiogenesis during wound healing. It can promote the tubular formation in ATCC Angio-Ready system too. These 6 representative images show with the increasing dose of VEGF, tubular formation efficiency is improved. This slide shows if you want to do high-throughput regenerative compound screening, you can do that using the ATCC Angio-Ready system. Again, we use a well-known VEGF as this compound as an example. Using a 96-well assay format, the tubular formation is responded positively to increasing dose of VEGF stimulation, which means if you have a similar compound, you can use the ATCC Angio-Ready system to screen it. Another example for using Angio-Ready system is when you want to inhibit angiogenesis, such as inhibit tumor growth. One of the most important ways to inhibit tumor vessel formation is to inhibit the tumor angiogenesis. So in the next several slides, I will use currently well-known angiogenesis inhibitors, including 3 clinical drugs which are used to treat the cancer patient, to show you how effectively the ATCC Angiogenesis system would be. The first compound we used to validate this system is suramin. We all know suramin is block the vessel formation in the very traditional way. So from these 6 represented images, you can see with the increase in dose of suramin up to 40 micromole, the tubular formation almost completely being blocked. Secondly, we used a clinical available drug. It's called sunitinib, which is marketed by the Pfizer. This is a research product. So sunitinib blocks the tubular structure growth in a dose-dependent manner. As you can see, you can see at the same time the anti-smooth muscle actin shows a tubular efficiency.
This slide shows sunitinib inhibits the tubular formation using the Angio-Ready system. At the same time, the anti-smooth muscle actin staining also shows the tubular formation efficiency is blocked by sunitinib. The anti-smooth muscle actin staining also indicates that the transformation of MSC to smooth muscle cells is closely associated with the tubular formation, which is another indication of the physiological relevance of the Angio-Ready system. We also do this in the real-time time-lapse videos. You can see the video from here in the 7 days time frame, show different concentration of sunitinib, how it performs in the ATCC Angio-Ready systems.
Sunitinib to 100 nanomolar concentration, almost 100% of the tubular formation is blocked. So next, we use another drug from Genentech to validate the Angio-Ready system in the high-throughput format. The left panels are the representative images, while the right panel is the quantitative tubular length data, which clearly shows the tubular formation is blocked by increasing dose of the drugs. So this slide serves for 2 purposes. The first, of course, we use another drug from Eli Lilly. This is monoclonal antibody can inhibit the tubular formation for the ATCC, the Angio-Ready system. And another thing is that you can see we use 3 lots of the Angio-Ready product, and 3 lots treated by this drug generates a very similar results. That echoes what Kevin has said. This lot-to-lot performance is very consistent from the Angio-Ready product. All those above data just point to one conclusion. If you have angiogenesis inhibitors candidates, ATCC Angio-Ready system will be a very robust tool, serves your screening purpose. This slide will tell you under another situation the ATCC Angio-Ready system can be used, which is hypoxia and tubular angiogenesis. We all know that during tumor growth, hypoxia stimulates tumor vessel growth. So if you have compound candidates which are targeting hypoxia, you can use ATCC Angio-Ready system too. This is a recently published paper on OncoTarget by Dr. Xu from Dr. Xiao's lab in the National Institute of Health using the ATCC Angio-Ready system screening drugs that inhibit the hypoxia factor 1. They even used the 3 days assay window instead of 7 days assay window, which gives even more convenient for high-throughput compound screening purpose. In this paper, they used ATCC Angio-Ready system validated 4 compounds which has been selected by other systems. And the data matched pretty well with the other several systems. And another news is I want to tell you that in the second paper is already submitted. They used the ATCC Angio-Ready system, screened the NPC library, which containing 2,800 drugs. A lot of using the ATCC Angio-Ready system, they screened a lot of known and new compounds from this library, which means that the ATCC Angio-Ready system is very useful tools to screen the compounds of interest. So with all the above slides, you realize that ATCC Angio-Ready system is really a good tool for regenerative medicine compounds, cancer inhibitors, and hypoxia pathway compound screening purpose. We will introduce other applications later on. Now with all of this, now I'm giving the control to our product manager, Kevin, to give you a summary of the product.
All right, thank you, Charles. All right, so I wanted to get everyone together and remember what our goals were when we developed this new angiogenesis assay. How can we develop an assay with better reproducibility, ease of assay readout, and make it more biologically relevant? And hopefully, from Charles's presentation of data from different applications, you can see that we did achieve all those goals. Using HTERT immortalized cells eliminates the concerns over reproducibility. HTERT cells exhibit primary cell characteristics with the benefit of continuous growth. The lines are derived from a single clone, so there's no lot-to-lot variability like you would see in primary cell systems. Using an endothelial cell that stably expresses GFP allows for live imaging of the assay during its run and an easy readout and the completion of the assay. Using a co-culture method of endothelial cells and MSCs instead of the more traditional fibroblasts allows for a more in vivo-like environment to develop due to the multipotency of the mesenchymal stem cells. In addition to the development of smooth muscle cells surrounding the newly formed tubules, the tubule formation is faster than in existing co-culture models using endothelial cells and fibroblasts. In the endothelial fibroblast model, it takes about 14 days for complete tubule formation. In the Angio-Ready system, the tube formation is complete in about 7 days. And as Charles has shown, depending on what your readout is, you may get enough tubule formation done in 3 days to run your assay. So in conclusion, I hope everyone has an appreciation that the Angio-Ready system does indeed provide an easy-to-use, robust, biologically relevant screening tool for angiogenesis processes. Thank you, and now we'll answer a few of your questions.
Oh, thank you, Mr. Grady. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. This session will be documented as a PDF and archived along with the recorded webinar presentation on the ATCC website at www.atcc.org. So this first question looks like a good question for Dr. Zhou. How can I quantify the efficiency of angiogenesis?
So most researchers quantify the efficiency of angiogenesis using the endothelial cell tubule length and number per square millimeter. Imaging software such as ImageJ or similar can be used to quantify tubular formation. For high-content screening, there is an automated image acquisition and analysis software available from different vendors.
Okay, Mr. Grady, can the angio-ready cells be propagated in culture?
No, the angio-ready cells are recommended for one-time use and should not be attempted to be subcultured. The reason for that, if you recall, the angio-ready cells are a combination of cells supplied in the cryo tube. It's a combination of H-TERT immortalized endothelial cells and H-TERT immortalized mesenchymal cells. They're mixed at the appropriate ratio for the assay to run, and if you tried to passage them and propagate the cells, one cell will grow faster than another, and very soon your ratio would not be appropriate for the assay, and subsequently the assay wouldn't work. So the cells are supplied in enough to plate into a 96-well plate, and it should be recommended is you would just use the cells in that format. You can put them in different size formats according to the directions that Charles gave during the presentation. But again, the cells are essentially a one-time use only for the assay.
Dr. Zhou, what markers of endothelial cells have been observed in Angio-Ready?
So we did a very typical expression such as endothelial cells, the CD31 and VWF has been observed in the Angio-Ready system.
Mr. Grady, once the VEGF supplement has been added to the medium, how long can I use it?
So stored in the refrigerator, it's stable for about a month. For longer-term storage, it should be at -20 in single-use volumes.
Mr. Grady, you mentioned the assay takes 7 days. Can the results be read sooner?
Yes. Depending on how the assay is set up, some researchers only go as much as 3 days, as illustrated in the data Charles presented from NIH. Complete tubule development in our hands, we normally recommend 7 days. But again, depending on what you're looking for in the results, the results can be read at any time point during the assay. Again, because of the live imaging, the assay can be read multiple times during the assay time frame.
Dr. Zhou, for your VEGF dose-dependent assay, why was there still a tremendous amount of tubular formation when VEGF was 0?
This is a very good question. We believe the reason could be two. First is the MSC secreted some amount of VEGF to the media for tubular growth. Secondly, other VEGF-independent pathway may contribute to the tubular formation.
Okay, and this is another question for Dr. Zhou. how was the hypoxia environment created in your system?
We believe the hypoxia condition is created by the overconfluent mesenchymal stem cells. The MSC feeder layer is very confluent by the time tubules are growing, which created a locally hypoxia situation which might be similar in solid tumors. There is some literature and the papers reported these overconfluent cells could create a locally hypoxia situation.
Well, at this time we will conclude our Q&A session. I would like to thank Mr. Grady and Dr. Zhou for the excellent presentation, and thank you everyone for attending this webinar. Any questions that were not answered this afternoon will be answered and posted with the video at www.atcc.org. Please join us for more webinars in the ATCC Excellence in Research webinar series. Thank you again, and have a great day.